Yes, sir. Yes.
Yes. So I'm sharing slides now. So let's see if you can see the slide. Can you see my slides?
Yes, sir.
Yes, great. So should I start?
Yes, sir.
Yeah. Thanks. Thank you again. Good morning, everyone.
Good
Okay,
morning, sir.
good morning. So I think this is again an online lecture. So one-to-one interaction is little bit less, but I still encourage you to maybe you can interrupt me in between. That's also fine. Or you can note it down the questions, and then at the end we can have a question answer session. I'll be happy to discuss with you because it's learning for me as well. So please do ask questions. So today I'm going to discuss about dereplication approach of finding new drugs. So dereplication, maybe some of you might not be aware about this particular terminology. So we will discuss about it and how it is useful in a natural product area or in also area where we refer is at a reverse engineering kind of thing, where we would like to know something which is there in the particular sample. So we will discuss about it. So before that, I thought I'll give you some background information about drug discovery and the contribution from natural product uh, research. So as you know that I'm not going to discuss all of these things in this slide. It's a crowded slide, but wanted to give you a message that it's a long history that natural products or the traditional medicines, it is from the plant based plant derived. Yeah. So plant derived molecules have played, I would say, the great role in curing many diseases or treating many patients, right? So, mm, even recently, we have the Nobel Prize in 2015 was awarded to Campbell and Omura and to UU2 for the discovery of ivermectins and artemisinin. And these are also great blockbuster drugs which are from natural product origin. So the take home message from this slide is that okay, natural product played an important role in finding new compounds, new molecules to supply the pipeline of drug discovery and development. Very important area. However, as I have highlighted here, from 1990s onward, there is a decline, a negative slope in the expenditure that the big pharma companies are spending on the R&Ds of natural product research. The reason being is that natural product research is relatively expensive. At the same time, the success rate is less, so companies are more interested where the logic fits properly and they expect more uh, uh, turnaround, so they want more and more success, and that's the reason the people have moved to the drug designing, combinatorial chemistry, parallel synthesis, and all these approaches have played an important or the major role in kind of declining this natural product research or contribution in drug discovery. However, nevertheless, that I would like to also mention that since then there was a limited number of new scaffolds, new entities entered into the clinical trials and in the market, and hence the recent conference which was held at Novo Nordisk in December 2019, uh, just before Corona pandemic, the big companies have also started thinking to spend more and to let's say start the process in the natural product drug discovery program, because if they don't do this right now, after 5 years or 10 years their pipelines will be dried, and so there will be no new molecules, new entities will be coming. So this is very important thing, just wanted to highlight that. And in 2019, one seaweed based molecule, actually it's a group of compound, they have been approved in China for the treatment of Alzheimer disease. So yes, the people have started working on it. There is a huge demand in natural product research, and there are several hurdles and bottlenecks. So that we wanted to discuss now, and how these dereplication techniques have overcome some of the problems associated with this particular program. So let's move on to my next slide. Let me, yeah. Okay. So in this slide, I am giving you a quick overview of drug development and discovery process. So for those maybe who are aware about this, maybe a repetition here, but for the total audience, I thought it is important slide to give you some background. So any drug which is like coming to the market has the companies are spending a lot of money on that particular molecule, and it is a time consuming process. As you can see here, it takes minimum 12 to 15 years for any molecule after its first hit, where we observe that okay, this molecule might have these biological properties. After this observation, if the molecule comes to the market, it takes maybe 12 to 15 years. It depends on the molecule to molecule. It depends on the how these particular different I would say steps, that how this molecule behave in that step, and then they filter it out or it goes further. So as you can see here, that there are different steps in the drug discovery and development program, starting with the lead of identification. Like I said, you can find out the lead or the hit from maybe a natural product source, or from combinatorial chemistry, or repurposing from metabolomics, and all drug designing using protein structures, all these things. So generally when you identify the lead, you also know the target of that particular lead, so where this particular molecule is going to bind. So I would also like to highlight the difference between hit and lead. So hit is the first incidence where you find out okay, this type of molecule or scaffold might have this activity. So that is your hit process. So let's say you have screened 10,000 natural product compound against, let's say, a cancer. Okay, and then you observe that okay, this is a particular molecule which has shown a nanomolar range activity, and that screening process is called as hit identification process. And then once you identify the hit, that is called as hit, then you modify that hit. Okay, it is not as it is you are taking, because some natural products or some kind of molecules that you are producing in the lab by synthetic route, they are not exactly fit for the particular biological activity. So then what do, you do the maybe in silico studies, where you try to dock that molecule on the particular target site, and then you modify that particular molecule to improve the efficacy and to reduce the toxicity. And that process result into, also you also try to improve the PK profile, pharmacokinetic profile, and that after that the molecule becomes a lead molecule. So hit to lead is a process, and that also takes some time. Then once the hit is lead is there, then you may go for the toxicity studies, preclinical studies, formulation studies, and all these things also needs to be carried out. That also takes a good amount of years, and then you generate a data for IND application. Okay, investigational new drug application. That application you need to generate a GMP or GLP compound, and that data goes to the regulatory and the FDA for approval. Once they approve that, you kind of start first in human trials, which are called as clinical trials. Then there are different phases of the clinical trial: phase 1, phase 2, phase 3. Phase 1 generally on the healthy volunteers, limited number of volunteers, where we check the toxicity of the particular molecule, safety profile of the molecule. And then in a phase 2, we kind of look into efficacy and toxicity on the larger audience, more than phase 1, and that also on the patients. Then in a phase 3 we go for the patients with a further larger audience from the different geographical locations to check the adverse or the desired effect, if it's any kind of population specific molecule and all these things you check. So this takes a good number of years, these studies, then approval and all these things. So after that you further file the NDA, for the approval to FDA, and then NDA is novel drug application. Well, all the studies and all the data is there in that particular application in the human trials, and that you submit to the FDA for their approval. And then phase 4 starts, where you do the marketing and early observation in the trial. So this is a long process, and the molecules that you start, let's say for example 10,000 or 20,000 molecules you start with as a hit, and then one or two drugs which end up into the market. So it's a long process. So we need to start with a good number of different scaffold rather than the similar scaffold in the lead identification process or the hit identification process. So this is very important part, and that's the reason the pharmaceutical companies wanted to focus on those molecule which will result into the success, and that's why they also started working on the me too type molecule, where they know that this scaffold has done good in phase 2 trials in some other companies. They prepare the use the same scaffold, they prepare the analog of that particular molecule, and then they go for the studies. That was some of the things that have reduced the interest in natural product. However, that's a short term success, not the long term, because in the long term you need a new variant, and you need a new scaffold. So, in the natural product drug discovery program, in the right hand side, I have shown one flowchart that remains the same. Only thing that differs is the hit identification. Here we do the hit identification. Yeah, so we do the hit identification from using the molecules which are present in the nature. Okay, now what happened, what are the major issues? So let's say now plant derived molecule has been studied extensively. Lot of researchers have worked on those particular uh, plant plant specimens, and they have reported the molecule. Now when you start working on the particular plant specimen, there are chances that you may end up with the same isolation of the same molecule. So that is one of the major problem. You spend 6 months to 1 year in this long process of isolation, like purification, then the characterization by NMR, XRD, IR, and all other spectroscopic technique, and then you kind of end up in saying that okay, this is a known compound, and that's one of the problem. Second thing is that the large scale supply, or maybe we can also refer as a sustainability method, and that also can be a problem with natural product from the plant based molecule. And then the identification of the target for this particular molecule, because at in the initial phases it is we don't know that these molecules will work through which target, and so the biological target, and that's the reason that also takes time. And so these are the major, I would say, the bottleneck in a natural product based drug discovery, and that led to the decline in natural product research. So to move on, I'm just explaining here again the problem statement in a great detail, because we will be spending good amount of time to on this particular process. So generally, when we take any plant material, we prepare extract from that plant material, and then we divide that extract in different fractions, so that we can get the proper activity data. And so once you fractionate it, you check the activity, and then that active fractions you further go out, and then you take the you isolate the compound, and then you characterize the compound, then you again check the activity, and then you get the molecule with the activity. And then if you realize this takes maybe 6 months to 2 years, depends on the molecule to molecule complexity of the extract, and then you find out that okay, these are the known compound, and so that goes to basically waste researchers. It's like a waste of energy, resources, money, and everything. So what if we can identify in that particular extract or the fraction itself that okay, these are the molecules tentatively are present in this fraction. So we can take a decision that okay, should we go ahead with this particular isolation and characterization process for this particular fraction or not. So this is the important question to ask at this particular step, because after that you are going to spend 6 months to 1 year on that particular fraction, and then you realize the problem. So it's always better to analyze that particular fraction, and this process of quick identification of the known compound is referred as dereplication. Okay, so dereplication is like the process or the method or analytical tool, if I say so, that identifies the compound, the known compound, a reported compound which is present in the fraction or extract. And this process takes hardly 1 to 2 weeks, and so it is very quick. Not in 1 to 2 week also I have considered a little initial extraction and all these things. Actual analysis takes maybe 1 or 2 days maximum. So it is a very quick process and identifies very easily a known compound, tentatively I would say, not maybe the 100% identification, but more than 95% confidence limit, and that's good enough to go and to take that decision to go ahead or to stop at that particular step. So we can use this technique to screen different fraction and focus on those fractions which can give you, which can lead to the novel compound, the novel NC, which is a need for this particular program. If you can observe on the right hand side, bottom of this particular slide, that from 2004 to 2013, so this paper is relatively old, 2015 paper, but I couldn't find the recent review article on this. So but you can see the message that the trend has been clearly seen, that the number of researchers, number of scientists working on this particular area, where the number of publications are noted here actually, has been increased tremendously. And you can see that it is there in the 2012 and 13, the bar diagram was like very high as compared to 2004. And so the process started at around 2003 2004, and so it has taken now a good amount of actually success rate. So the in this bottom left side I am showing you the, just 1 second. This diagram where it says that natural product resource, bag of tricks. So these are the nothing but different dereplication approaches that you can use it to early identification of the compound. And then if the compound is known, you do not work on that particular extract, you just keep it aside. And if you find out okay, no, there is a novelty, the molecules might be novel in this, then you move it to the that particular next step. And so the chances, I would say, of finding out novel compound from that particular extract increased tremendously. So I hope I made that particular statement and the particular problem statement very clear, and the use of the replication technique. So I just want to put the definition of this so that students will remember this. So it is a process of testing samples of mixtures which are active in a particular screening process, so as to recognize and eliminate from considering those active substances which are already studied. A stage subsequent to the preliminary screening in the process of discovery of new pharmacological active substances in the mixture of natural products. So as we discussed earlier, it's the same thing, just in the definition format. So it is a process. It is not the way that you will end up with isolating a compound, but it is a process. It's a testing tool which can help you, which can guide you to select or deselect a particular fraction to go for the isolation and characterization. So let's go move on to the like in detail about what are the different methodology, what are the different techniques or at instrumentation these are being used in this particular method or the dereplication process. So, like I said, it is a quick identification process, and we use advanced instrumentation here, which are the sensitive enough to find out the small quantity of some compound in the particular extract as well. So that's why the methods used here are mostly the hyphenated methods. Hyphenation means the combination of the two major techniques, two major analytical techniques, which independently also can work, and in together they work as a method which gives the additional information. So, for example, if I say LCMS, LC can HPLCs are independently can be used. Mass spectrometers also independently can use, but in LCMS together they can give much more information that independently these two machines cannot give. Okay, so the chromatographic part, which is a HPLC part here, LC that I have mentioned here. So these parts are used to separate the compounds in the mixture. So let's say there is a mixture, there is a natural product extract or fraction containing 10 molecules, 10 compounds. So these part will separate those 10 molecules into at a different RT, these different compounds will elute, and the mass spectrometer will analyze that at different times and give you the information about the molecular weight, accurate mass, fragmentation pattern, and all this information you can get it through the mass analyzer. There are also another method we refer it as LCNMR or LCMS-MS-NMR, and there are many more. But LCNMR and MS-MS-NMR is also being used where you can connect the NMR also uh, attached to the LC, and that gives the information of the live compounds which are coming out of the column to the that you can monitor it through the NMR. So we will mostly focus on the LCMS part, and because which is the highly acceptable due to two reasons, that LCNMR is quite expensive and challenging as well, because the NMR has a poor sensitivity as compared to MS, and the LMR also has an issue with the deuterated solvent, because we have to use a deuterated compound to get a proper NMR, and they are expensive, and so that's the reason it is a limited use. It has a limited use. But LCMS, LCHRMS, LCMSMS is like widely used methods for this particular application. So now I want to also mention one thing. It's not only the analysis that is important here. We also need a database. The database means a collection of the data which is already reported, right? So what we are doing here, we are trying to identify the compound which is already reported in the literature. How will you know that if it is already reported or not? By looking at the database. So that's the reason databases are very important in this process. There are several databases available, n number of, and I am putting here only few that maybe I have come across, and it's available, and they are kind of uh, having a very large number of molecules in this database. Okay, so like first was the antibase, where most of the microbial origin molecules are included here, mostly antibiotics. Marine lit is related to the molecules which are obtained from the marine environment. NAP alert also related to natural product, and dictionary of natural products is also there, which kind of combine all of these and some plant based molecules as well. So dictionary of natural product is kind of comprehensive, which contains more than 3 lakhs molecules, uh, other than like fatty acid and all other things, the specific molecules, and these are there in this database. Then there is a SciFinder also, which can be used to do the structural search to find the novelty if the particular molecule you have isolated, and then the structure is, let's say you want to check if it is already reported or unknown. So that can be used. So these databases are also very important in this particular step where we check that okay, this molecule is already reported or not. What is the important part of this database is that it can be you can search within this database. So for example, you have a big book talking about all the molecules coming from natural resources like the dictionaries that we earlier we used to use. Then to finding that particular molecule or the name in the dictionary is a little bit difficult, like as compared to Google search, right? So nowadays what we are doing, we are just putting it into Google and you get the detail very quickly that okay, somebody has already worked on this. Like in YouTube also we try to find out okay, any video is there related to this particular topic, correct? So same thing, I think you guys are more expert than me in this process. So using internet as a tool, so same applies here also in this database. In this database you can search, you know, like for example in case of dictionary of natural product, if I want to know how many molecules are there with the molecular weight 370, if I want to ask this question, I can just select a molecular weight option, add 370 and search. So out of 3 lakhs, 4 lakhs molecules, it will show you maybe 10 or 20 molecules which is having this particular molecular weight. And then you can also further filter it using the biological source. For example, I want to find out in the neem tree. So I can put it genus Azadirachta in there. And so all these things you can do and just modify or narrow it down your search and find out, okay, if this particular molecule has been reported from this particular plant or not. And this is a use, this is a strength of this particular database. It is not only the molecular weight, but it will also have the UV profile, melting point, and a lot of other things are also there, like accurate mass information. So very, very useful tool. So in the right hand side of this slide, I am showing you one particular flow chart which is mentioned in this particular paper, that how people are going forward by applying this dereplication technique to find out a novel scaffold. So ultimately, all of these things are to find out novel compound for drug discovery, right? And so ultimately, we are looking for drug discovery, but it is a starting process of that basic research, I would say. So when we do the fractionation of crude extract, these fractions we analyze with the dereplication tool to find out, okay, if any new compound is present or not in that particular fraction. If it is, then we screen for bioactivity. Then we kind of enrich that second type of screening and isolation of bioactive compound. Now imagine a situation where I have removed this dereplication process from this step and I have added into the third step. Instead of second step, I put it in the third step. That's a different strategy altogether. So in the first strategy, what we are trying to see that, okay, if the compound is novel or not, I want to check that first. And then I want to check the activity. In the second strategy, what I want to do, I want to check if the fraction is active or not in the respective biological assay that we wanted to work on. If it is active, then I will go with the dereplication to find out that which type of molecules are present in that particular fraction. Now, when we know that, okay, let's say I have checked anti-cancer activity and the molecule, I found out that, okay, this molecule X is present as a majority and might be responsible for the activity because it's the major compound, not necessarily, just a hypothetical situation. Now, I will check in the literature that compound, what are the different activities reported for this particular molecule. So for example, it is reported for the antimicrobial activity. Now I have a novel bioactivity of that particular compound. So maybe I would go ahead and isolate that compound and get the activity. So there are two output of this particular process. One is called as New chemical entity, NCE, new chemical entity, that is a novel scaffold, novel structure, new activity. It's a bioactive compound, not the without bioactive. And another output is NCA, so novel chemical application. So the compound or the structure is already reported, but the application is different. Both are important in drug discovery arena. Okay, so that is the process. Most of the time, people are following in the dereplication media, and I think I discussed about this slide earlier, so I'm just keeping it out. The application, just for the audience here, because we do not have students from the pharmaceutical background, so it is not only the drug or drug related compound. This can be applied to the another areas also, not necessarily the pharma area. So it is for the microbial metabolites. You can check which are the metabolites microbes are producing, and that can be useful for the any other application that you want to check the leaching or the there are now a lot of regulations are there, leachables. Like let's say if you are storing a particular product in the plastic container, and then you want to know that during its lifetime or shelf life how much that plastic is released into the particular solution. You want to check that which are the compounds are there, then how much quantity of those compounds are present. That can be easily done with this type of technique. Okay, and it is only not related to this, but you also can be used in the food application. This is one of the paper that I could get that in that they are talking about using HPLC, DAD, ESI, TOF, MS, and I'll talk about this. Uh, and they have done the metabolite profiling. Which are the metabolites present using the replication based approach in the sugar maple bark. So these are the different applications you can use. So this is nothing but again the LCHRMS. This is HPLC. I think you maybe you might be aware about high performance liquid chromatography. DAD is a diode array detector, where it is mostly we are working on the compounds which absorb the light in a UV visible range, and that's the chromatogram you get here. Then ESI is the ionization source, electrospray ionization. TOF is a mass analyzer, which is the time of flight, and MS is a mass spectrometer, the name of that. So this is nothing but the LCHRMS, that one of the technique that we have mentioned in the earlier thing. So these type of mass spectrums you get, actually this is a chromatogram, and then these are the different. Chromatogram means you have a compound, you have a illusion pattern which is coming out from the column, and you're monitoring it with UV or DAD or maybe mass, and that's the chromatogram. Basically, you have a retention time for different molecules, and then you identify those molecules which are present at a different retention time. So it is not only the pharma based application, but also the other application where you can apply in n number of areas. I think there is no limit to it. So the very good application. So now I will little bit switch gears, and till now I was explaining you the method, which is I would say started in 2002 or around that time frame, and now it has taken a good acceptance. Actually, it is accepted by a lot of scientists all over the world, which is called as dereplication method. Now, I will in next maybe 15 to 20 minutes, very quickly, not much. I will share some of my experience on this particular methodology, how we have applied in our labs, and not only in NIPER, but also in the previous organizations where I work. And uh, I will show you some outputs or the results, that how it was useful. So that the real-time examples, not only based on the literature, but on the actual experience I will share with you. So fortunately, I came across the various natural specimens in during the research, and not only plant but also from marine invertebrates. Like here I am showing you the coral, which I have worked during my PhD program, and I have also worked on various microbes, and that is also very important natural source nowadays, because we cannot collect plants indefinitely. We are disturbing environment, and we do not want to do that. For marine and ecosystem also, we cannot disturb in the large range. We can take a small quantity, you can study in the lab, but ultimately when we go for the large scale production of this particular molecule, we cannot rely on nature for this particular thing. So that's why these methods are not sustainable methods. These methods are not like environment friendly methods. So we want to go for somewhere some natural specimens which we can cultivate in the lab, and we can depend on them, we can grow them on the larger quantities, and that is nothing but the microbes. And so microbes have a tremendous potential to produce a diverse secondary metabolites. In fact, scientists have studied the microbes residing in the plant. They are called as endophytes. Endophytes are bacteria, fungi, all of this are there, the endophytic bacteria, endophytic fungi. They are also residing in a marine specimens, and these uh, some of the hypothesis is that these microbes are actually the producers of this particular secondary metabolite, because they are producing them in the nature for their survival, and that is what we are using it for our own benefit. So why not study microbes directly? And that is what the trend is right now in a natural product area. Like I said here, it's isolated from different specimens like soil, plant, marine sample, water, sediments, invertebrates, and a number of area. Like currently we are working on the endolycanic fungi. So the fungi which is residing inside the lichen, that we are studying to find out what kind of molecules they are producing. And so it is very interesting to see that because the biodiversity resulted in the chemical diversity. Now, to study this, we use a lot of instrumentation, and which makes our life relatively easy, because otherwise to collect the fractions, to collect the samples, somebody has to sit there and collect all manually. While these auto samplers and liquid handlers, this was the liquid handler which was present in my lab earlier, and this we can even inject something onto the column. At the same time, we can use it for the collection of the fractions. So like you can see here, different pixelating at a different time can be collected at a different tubes. And then we can also put here some of the 96 well plates that can be used to screen the bioassay. And then we can even check the UV profile, and if they're mass based, because one of our method was mass based. I have a slide on that also, that we will show you, so that we select the fractions that we want to work beforehand by checking the dereplication index. So this is the phase one, what we call that in the drug discovery, not the clinical phase one, but in the discovery, which is called as nothing but a dereplication technique. So this is a real picture that was present in our lab. That's why it looks not that much polished one, but it's like the photographs of the actual instrument that we can this is the HPLC column compartment, and that column I'm going to show you this 6-foot wall. Then there is a auto sampler here, a pump, and solvent chamber is on the top. This is a DAD detector that I was talking about, diode array detector. Now, which is this is a non-destructive detector, so you can put all the samples through that. And then we used to divide that sample in two parts. One is used to go in the mass spec, and the another one for the fraction collector, like this, to check the bioactivity. So we check the bioactivity parallelly, and we check the databases and mass analysis parallelly. Now we know that, okay, this particular region here has shown a good bioactivity, means the compound which is present at eluted at this particular time has shown the bioactivity, which is we used to check it in the 4 different area: antimicrobial, anti-diabetic, anti-cancer, and anti-inflammatory effect. with the different IL-6, TNF and all. And that we can compare with the mass spectrum data and check that, okay, if that compound with that particular activity is already been reported or not. And in parallel to that, we this one of the project was there as was the DBT sponsored project. That we screened 700 microbial extract. Okay, and there, the microbes, different microbes, though they are different, collected from different area, they may produce a similar profile sometimes in the same environment. And so we used to club them. So cluster analysis, chemical fingerprint, what we used to call. So chemical fingerprint is nothing but to see what are the different chemicals they are producing. So here we used to use three detector: ELSD detector, UV is nothing but a DAD detector, and mass spec detector. So this is another instrument, and this was UPLC, not HPLC. The difference between the UPLC and HPLC is UPLC is relatively faster than HPLC, and you get a similar separation in a shorter run time. I know the time is running fast, so I'll quickly go through the next thing. So here, basically, we used to club the similar fractions or the similar microbes which are shown the similar profile together and select one or two from that particular microbe. So that was the first part that we select, and then we go to the next step where we do actual purification. So this is like the next step where we develop a method for this is the analytical method for the separation of the compound. You can see the complexity of the fractions. There are so many compounds, minor compounds present here, and some of them are the major compounds in that particular fraction. So this is PP means nothing but the plant origin extract, and that is that I'm showing. Then we kind of enrich that particular fraction. Yeah, sure. So this fraction, this fraction we divided into the different this extract we divided into different fractions by using the vacuum liquid chromatography, and wanted to check the which area has shown the novelty as well as the activity, and we observed that this 40% fraction has shown the novelty as well as activity, and so we move ahead with that. We developed the HPLC method to separate those compounds. Then we move for the preparative HPLC. In the preparative HPLC, the flow rate was 100 ml per minute. Generally, in HPLC, we use flow rate as 1 ml per minute. Now, this is 100 times more. So you imagine that if you have a bottle of 1 liter as a reservoir, in 10 minutes, that bottle gets over. So the reservoir was also like 10 liter or 20 liter reservoir was there in that. And we use a column which was 50 mm. So it is like a big ID column, not like a very small column. So we can load a good quantity. So around 1 gram of the samples we could load on this particular thing, and we collected these compounds as I have shown, which was relatively pure. One of that I am going to show you the structure illustration, not in the detail, but just a quick overview how generally we do it. And then you get the mass information, and then we check the purity, UV profile. As you can see, the purity of the compound is greater than 98%. We checked at the different chromatography. We checked at different detectors as well. Then we checked at our database that okay, it's MS and NSMS fragment. Does they show any similarity in the database or not? This is a total ion chromatogram of that particular sample. And then we, after purification, we moved on and we perform a detailed NMR study, which is nothing but a 1D and 2D NMR and all the other related study associated with, and we identified this as a novel chemical entity. It's a new compound that later on we have published also, because earlier we have tried to patent it, but then the activity was not that great and the company was not interested in that particular area, so we published it in the a good journal in that day. Now, I will talk about some of the quick. So this was the process I have shown you how we work, and I will now share quickly in one slide that okay, how many compounds we dereplicated in early phases, and how many. That's why we led it resulted into the identification of the fraction, which resulted into the novel compound. So, for example, if I talk about this coral, which is Pseudoteres gorgeous serosa, this coral already when I started working on that, already 24 compounds were reported from this coral. But I could also found out 9 new compounds from this particular coral, which were published in Journal of Natural Products. So we dereplicated lot of molecules from this particular coral. However, we found out new molecules also from this. And in fact, we first time we reported a lipidyl pseudoterene where the pseudoterene ring was attached with the lipidyl fatty acid chain, and that's the observation that the first time we reported. So this is really an important tool to study. So what was the difference here? Earlier, people have used only bioassay guided fractionation method for this particular coral to get these molecules. But then later on, we have changed the view, and so we have changed the technique, and we end up with the 9 new compounds from this particular coral. So that was the thing. So we need a new actually approach to study different natural specimen, and we may end up with getting in the novel compound. Now, this is a recent project that we were working on. We are working still on some mangrove based lichens. So we collected lichens which grows on the mangrove plant, and this is one of the mangrove plant, Avicennia marina. And we collected from mostly now we have collected in a limited area, but we want to explore all the region of Gulf of Kutch, where the Marine National Park is there, and Marine National Park, and nearby that area also. So our idea here is, see, this actually mangroves grows in between the ocean or seabed and the land. You know, it saves the land for the erosion, and but that soil, the water is very salty. So and there is humid condition, uh, and hot also. So extreme weather is there. It's not easy to survive there. Our regular plants also do not survive in that particular region. Now, with that extreme environment, lichens also are growing in that those particular. So these lichens are the ecosystem where like you have a lichen forming fungi, then you have some symbiont, which is mostly algae or cyanobacteria, and then you have some other microbes residing in that, like I said, endophytes, where the plant microbes residing into the plant, like the same thing, the microbe residing in the lichens are also there. So uh, we directly wanted to go and look into those microbes, because you know we want something which is sustainable. We cannot collect lichens all the time to get these molecules, and they are also very tiny colonies, so you cannot really depend on them for the large scale. So but they produce a diverse compound in the Indian, if I talk about different lichens present in India, there are so many lichens reported from India, but very few are studied in terms of the chemical diversity. And that is what our aim is to check. This is also a DST funded project. It is between the collaboration between India and Sri Lanka, because in Sri Lanka you find these lichens in abundance, and we visited Sri Lanka, and we are working on some of the lichens from Sri Lanka also here. And so we have identified, like I said here, also we have dereplicated several molecules. So microlactam ether, some small molecules, also a very small molecular weight, and we also got some NC as well from this. NC is a novel chemical entity which is showing a relatively decent anti-cancer activity. I would say around 3 to 4 micromolar in breast cancer cell line, colon cancer cell line that we have observed. So really a good start to go with, and Oh yeah, so this is actually the lichen look like. This is one of the lichen that I've collected. Actually, this lichen was collected from Rajasthan area, and this from this lichen we started this particular culture from fungus, and so this is the endolycanic fungi that we are referring it to. And we have also worked on some several lichens collected by our collaborator from University of Kelaniya, Sri Lanka. And this NCE, actually this NCE is from this particular from Udaipur region lichen. But yeah, we have got some good structures also from lichen which is collected from Sri Lanka. This is about the data that I have shared. Now I want to give you how we are working on this LC HRMS part. You know, what is HRMS? Because at the end of this presentation, I think that you should at least know and you should at least appreciate the methodology and not the data. So I want you to remember the process. So like when I say LC HRMS, so HRMS is a high resolution mass spectrometer. So high resolution is like nothing but our analog clock and digital clock. You know? So we can get accurate information with a digital one. And the same applies here as well. So as you can see here in this slide, there are four molecular formulas which has a molecular weight 44. So if you can read only 44, which is actually the case in a low resolution mass analyzer, then you cannot distinguish between all these four molecules. You can just say that okay, it can be one of this. But if you can identify accurately, it is not 44, it is 44.029. Then you can pick up the correct molecular formula, because you know each of these atoms have a specific mass. So for example, carbon is 12.00 that we have fixed it, but the proton is 1.007824. It's not 1, it's 1.007824. Same as the oxygen, 15.9948 or 44, something like that. So same thing. If you add all these things, you will get accurate mass information. So for example, in C3H8, the accurate mass is 44.0626. For this molecule, it is 44.0262. For this, it is 43.98. For this, it is 44. So our observed mass is 44.029. So it is close to this particular molecular formula. And so that's the reason we can pick up, okay, this might be the molecular formula of a particular compound, which is very important for us to identify in extract that, okay, this can be a molecular formula for this particular compound. Same application here, though the molecular formula is 60 for nominal mass is 60 for these two molecules, it is even the precisely, if I want to say, they are different, and that can be distinguished with a high resolution mass spec. So we use high resolution mass spec data for this particular experiment, and that's how we select and we identify the molecule at early stage. We are also working on a different plant here at Parambaba. Just wanted to give a glimpse of it. This is a Kachunar Baunia variegata, Alstonius fluorescens, which is nothing but a Saptaparni devil's tree, and we are specifically working on these gall tissues, which we observe very commonly. You might have observed also in any garden or anywhere that this type of galls is on the, they are on the leaf of this. So we wanted to see what kind of chemical diversity is there in that. Then this is also an interesting plant, Nicostema laterale, where we wanted to work on specia marine. And then this is a neem tree as a replica. There we have established this some of them reported molecule, and we have dereplicated with this product. So this is one example. I'll take 5 more minutes and then I will stop, and then we'll go for a question and answer. So this is also one of the study related to the plant, which is also important to study. This is a Kachunar Bahunia variegata plant, and one of my master students have worked on that. And what we have observed here, interestingly, when we analyze it in the extract level, that you can see it, there is a peak which is at around 35 to 45 regions, and which are like present but relatively there in the GCM methanol extract and ethyl acetate extract. These molecules were present. We have dereplicated this particular some of the compounds from this particular plant, but we also observed some of the compounds which in the plant with no hits in the database. Now, I also wanted to highlight one thing here is that here. So we fractionated it earlier with a column chromatography, which is a common technique in the natural product lab, where we use a column and we fractionate it in the different fractions so that we can enrich some of the peaks which are present in that. And then what we did is when we enriched it, we have further performed this analysis at a different wavelength, in the EFPD detector. And you can see that these particular class of the compound have shown peak at all three wavelengths, while the other peaks are not there in the other wave. So this is a class of the molecule that are these are maybe the group of the molecules from the similar having the similar scaffold, and then we check the masses and all these things and the uniqueness about this scaffold. We observe that these are due to the phenanthroquinone type of molecule. And so these molecules, very few molecules are reported from this particular from plant from this class. So we went ahead and we isolated this particular molecule like preparative HPLC. We purified it with a good purity around 96%. And we checked the anti-cancer activity. We observed it's like very good. Doxorubicin was a positive control. And we observed that it has a zero point 0.1 micromolar anti-cancer activity, this compound. The structure elucidation, by the way, it's still in progress. We still sorry. Excuse me. So this is a like I said, accurate mass to understand the molecular formula, and we think based on our current observation and based on the accurate mass information, some kind of NMR information that we have, that this molecule has not been reported in the literature yet. And so this is most probably the novel compound that we will get from this study. So again, I would say that these methods are very important, and we should uh, employ them to get a better success rate. Sometimes we also end up with getting the known compound. That is possible because there are also some of the exceptions are there. And however, the probability is more if you apply this technique. So I I'll stop here. Before conclude, I want to acknowledge my students who have mostly worked on these projects, and this project today I have shown you the Ashwini's project, that the data. There are PhD and master students that we have in the lab here. I also would like to acknowledge our director, Professor Kiran Kalia from Ahmedabad, for his support and the funding that we received for this particular project from Ahmedabad as well as from DSP. This project. This is actually my last batch and some of the PhD students. Uh, I'll stop here and I will be happy to answer any question if you have. Thank you so much. Hello?
Hello? Yeah,
Yes.
students. Now you can ask your questions.
Yes.
Yeah, 2 to 5 minutes for question and answer.
I hope it was not too long.
No, sir, no, sir.
No question?
Shweta, yes? Hello?
Your voice is not audible.
Hello? Her voice is. Students, any question, any queries, not only for this, you can ask about Nightbar and what Sir and his group is doing at the
You can
institute.
ask about anything that you are working on and you may apply this methodology to solve some of the problems associated with it. That is also, I will be happy because that is more learning for me as well.
Yes, and some of you have also selected the plant-based grassroots practice, so you can ask about that too.
Yes. So here we have in our lab the dictionary of natural products. So generally we use that, like I said, as a database to identify this compound, which is like unique and we can identify like, okay, how many molecules from this particular plants are already reported so that we can focus on that particular thing. If it's interest.
Yeah. Guys, any question? Yeah, Sweta, you can write in chat.
Yes, you can write in the chat box also. Or if you have any questions, you can write to Megha also and you can forward it to me in future and we
Yes,
can
yes.
discuss about that also. No problem.
Sir, thank you so much, sir. It was very enlightening, a very new topic for me personally, sir. So it was very inspiring, sir. Just a few questions, sir, about the regulatory aspects, sir. So we have our DG, that is, there's a drug control body in India, and
Right.
that's FDA, right, sir? So generally, when they talk about any allopathic medicine, they say it is FDA approved or I'm not sure, but I think they say DGCI approved or DCGI. I'm not sure,
Yeah,
sir.
it's a CDESCO body that approves the drug in India, which is headed by DCGI. Yes, Drug Controller of India.
Sir, so how important is it to get FDA approval for such kind of plant-based medicines that we have created, sir? Or is it even possible to get FDA approval because, or is it needed, sir?
Very good question. Thank you for asking. Actually, I don't know the exact answer, maybe to be honest, right now, because the answer doesn't exist as it is. There are, I can answer in the maybe a probability way. So two ways are there. Okay, the one way is you can develop a molecule from plant in the allopathic medicine way. Okay, where which will take it as a same route that any new drug develop. The another approach is using as a herbal. You know, in herbal also there are two chances. Either you can go with a Ayush medicine, which is like Ayurveda based, and for that you need a reference in Ayurveda for that particular plant. But the another approach is relatively new, not new, but relatively new, is called as phytopharmaceuticals. In a phytopharmaceutical is approved by DCGI again, like the our regular allopathic medicine. Okay, so this is approved by CDESCO, not by Ayush or any other Ayurvedic thing. So this is like more standardized method of using plant extract, but there are some requirements. You have to have a minimum four markers that you should identify with the quantitative data you have in all your formulation. Then you should also carry out some clinical trials for that particular formulation. It is not as extensive as our drug, which is a pharmaceutical product, but it is there. Not that extensive, but yes, up to certain extent you have to perform. So there are several plus and minuses here. Like in herbal medicine, you have advantages that you don't have to carry out so many experiment or trials, but you need a reference in the Ayurvedic text, and people have consumed it earlier that you have to show. Okay, but while in the other case you have to identify the marker, identify the you have to perform some of the processes, toxicity studies, and all these things, and then you have to prove it in the humans also. But the advantage of those studies is that it is a well accepted now formulation, and that now you can export it. It's worldwide acceptable. It is not only in India now. Remember. So that is a plus and minus for all the products which are coming from the plant. There is one more concept, is called as nutraceutical or functional food. Right now that is also coming out very much, and people are taking it. But that is also something you may need a standardization there, like some of the companies are working on it. But the regulations are not that much at this stage, but they are coming in place. So I hope I try to answer your question, but there is no one and quick bullet for this, but there are different options that you can go ahead with the plant derived molecules and the extract.
Yes, sir. Thank you so much, sir.
Yeah, sure.
So there are a few questions in chat box.
Chat box. Okay. I want to chat box.
Can we make any drug naturally?
Here. Yeah. Can we get naturally and make that composition? So the question is, can we make any drug naturally and mix that composition with artificial drug composition? So what I understood, I'll repeat, and then maybe you can confirm the question if it is right, what I understood. Is that can we find out a molecule naturally and then make that in the lab with some other technique? Is that the one that you were trying to ask? Yes. Okay. Good. Yes, definitely. That is that is the method actually that we are also following, because, like I said earlier, we cannot rely on plant for producing this molecule for us, because we cannot cut the trees like that. So what we do is, with the research purpose only, we work on that. We identify the scaffold, we identify the structure, because human mind are very have limited thought process. We cannot imagine something which we have not seen earlier. So we need we kind of only modify something, but not completely different we can think about. But the nature can, and so that's why we get inspired from this, and then we synthesize it in the lab. So most probably we do the synthesis in the lab, and then we take it as a forward for that particular molecule. So that synthesized one is a artificial one. Now another approach is there. What we can do is we can do the semi-synthetic also, where we synthesize, we take the microbes which produce the scaffold, then we use that scaffold and modify it to get the desired molecule. So there are two methods. One is synthetic total synthesis, and another one is the semi-synthesis. So I hope I answered your question. So next question is, good morning. How to do metabolite screening using LCMS in lipidomics? Okay, great. So there are two, I would say, parts in this. One is targeted, and another one is untargeted. So targeted lipidomics, targeted metabolomics, where we are looking for those metabolites in that particular cycle that we expect, and we are checking the abundance of those, quantitative analysis, like which one is upregulated, which one is downregulated. Yes, so that can be done with LCMS, and for that we generally use a triple quadruple instrument, and that is kind of MRM methods are there in that, where we have different MRM for the different metabolites, and we run our sample, like generally control and maybe disease or any particular set of the sample, and then we check the differences in that, and that is how we do the targeted metabolomic, targeted lipidomic. Lipidomic mainly we use the GC triple quad. Metabolomic also we use the GC triple quad or LC triple quad. It's based on the The molecules, if they are small molecules, we go for a GC, and if they are large, then we go for LC. Okay. Now, untargeted ones. This is little bit complex. Untargeted ones means we just do the random. We check all the things which are there in the particular uh, spectrum or the mass HPLC data. So there are so many peaks. So we need to go one by one. So we acquire the data in maybe 4 to 5 hours, but we analyze data for maybe 3 to 4 months. They're huge data. We try to compare it. There are different databases also available in the literature like Uniprot, Chemprot, Swissport, all these things where we kind of check, we import those and we check it in the, there are different softwares that we use. Like what we have is a spectrum mill software where we can check the homology with the matching with like different protein sequence we match with the reported one what we observe, and that gives an idea about, okay, this protein might have been upregulated in this particular situation. And that is a little bit tough task, it's a long process, but the targeted one is relatively simple and that can be done very easy. So yes, this is definitely a new area and a new thing where the actually the pharmaceutical companies are also moving towards the uh, let's say biopharmaceuticals, biosimilars, biologicals, and all these things where mostly the proteins and peptides are being used for the treatment purpose. Yes, I hope I answered this question. Like, can we make a drug from particular plant? So that I think I have discussed that. So next question is, it's a general question, but why there is always a conflict situation between allopathy and Ayurveda, homeopathy, though it's almost same or similar compound acting to the disease? Yes, so conflict is like human mind always look for the logic. When we don't find the logic, we don't accept it as it is. This is common thing, right? So that's why the conflict is. In allopathy, everything is black and white. We have proved it in allopathic that this molecule, if you take, it will bind to this and it will produce this effect within this time, right? And that is what it is well accepted. It is overall in worldwide people are taking it, and we know the side effect also of that. So all proved experimentally in the lab. And in Ayurveda and homeopathy, we are lacking some of the data. I am not saying that it is not effective. It is effective, maybe sometimes better. And like there are in diabetes, for example, we are also working on one of my projects. So uh, we can just kind of, we cannot treat or cure that particular thing. We cannot, it is very difficult for us to increase the beta cell mass to which helps to produce the insulin in some in patients. But in Ayurvedic formulation, some of the studies have reported that it is basically increasing the beta cell mass so that you can cure that disease also in long term if you take that. So that's what we are studying right now in our natural product group. That what are those? So our job is now to bridge this gap and fill this gap and make it proved scientifically to all the audience to make sure that we get a proper acceptance all over the world, not only in India. So we in India we trust, and so that's why we accept it and we consume it. But in a Western country or in other countries, I would say we why would they trust to us? We have to prove that, okay, we, if you give this, this will happen. And once we do that, I think that conflict will go away. I guess I can give a little bit answer, but yes, there is also some other arguments might be there, but try to put out my view on that. Yeah, any other question? I think I have gone through all the
Sir,
questions.
one question, maybe
Yes.
it may sound like different, but what do you think that in the current science there are lack of any technology or any theory which can prove the Ayurveda's theory? Like, see, it may be the we can we say this कि हमारे पास ऐसी कोई टेक्नोलॉजी नहीं है जो उस मैकेनिज्म को एक्सप्लेन कर सके, या ऐसी कुछ थ्योरी नहीं है. So can we say that like it's yeah, out
Yeah.
of like something, but
No, very good question. No, there is no question which is out of, because this is always learning, and that's what I like about these sessions, because we get a different perspective altogether. So there are two approaches, Mukesh. One thing is that people are referring as the reverse pharmacology and forward pharmacology to prove the Ayurveda thing. So reverse pharmacology and forward pharmacology, like I said. So in one case, the target. See, target identification nowadays is very important. How that molecule, how that extract is working, we should know that, right? So it's simple, right? We buy so many gadgets and all. The first thing we think about, like, कि कैसे काम करता है ना, ये तो पहला क्वेश्चन आता ही है दिमाग में, बराबर. So we need to know that, and that is our main job. So that's why nowadays in silico methods are there. We also are using. We have Schrodinger software in our lab, and so in silico data, what we have is we have different targets in silico. Actually, we do not have to purchase anything. It is there in silico, and what we can have is we know the different molecules present in that particular extract, like we are using in Ayurveda, and that molecule we are trying to dock. Docking means we are trying to see that how best they fit in that target, and that is how we are trying to find out the target of this particular molecule. So in that's a good question you ask. In our lab, we are trying to find out some of the interesting molecules, like I was talking about Anicostema litrole, that plant extract people are taking for the diabetes treatment, and it is really useful. uh, but the formulation doesn't work properly, but the fresh extract really works well. So we are trying to find out what's happening with that particular molecule, which molecules are there, which will work, and how they will be, how they are working to get the desired effect. So that is our one of the project. The one of the master students are working on that thing. So there are different approaches like reverse pharmacology, like I said, where we first try to find out the target, and then we do the enzymatic assay, where we see that actually that molecule is binding to the target and giving the effect, and then we go for the in vivo study. Ultimately, we have to prove on animals that okay, it is working like this, and then we can prove that okay, this Ayurvedic formulation is working through this target and giving this effect. But with that, we will know the markers, and then henceforth, we will have a method of standardization that any batch coming with that particular plant should have this molecule, this concentration, so that you get a consistent effect. And that is what is needed right now, I think, to standardize the this particular formulation. I hope that is what that's the answer of your question.
yes sir, sure, yeah,
Yeah, good.
yeah. Thank you sir, thank you.
Thank you. Any other question?
So I guess there are no more questions, sir.
Okay.
But anyway, sir, if I'll receive any question, I'll definitely forward to you.
Please let me.
And thank you so much for your time, and it was really wonderful and interactive session, as always.
Thank you. Thank you
Thank you, sir.
again for giving me this opportunity to speak. Thank you.
Yes, sir. We look for your support for such future workshops, sir.
Definitely,
And
madam. Thank
hope to see you.
you. Yes,
Yes.
in person. Yes, definitely.
Yes, sir. It's very important now.
Yes, yes, yes.
Okay, sir.
Okay, everyone. Enjoy and take care. Thank you.
Bye,
Thank
sir.
you sir.
Thank
Thank you, sir.
Bye.
Thank you,
Thank
sir.
you sir.
So
So guys, now we're going to discuss about your grassroots practices you have selected. Wait for a minute. Hello. So, am I audible?
Yes, ma'am. Yes, ma'am.
Okay. So I hope आप सब लोगों ने अपना grassroots practice select कर लिया है. And I can see here your Excel sheet. I'm sharing this Excel sheet. Can you just see quickly if some other students have selected the same practice? Because if I'm alone going to see this, it will take time. Can you see this now?
Yes, ma'am.
Yes, ma'am.
Yeah, so there are no reference ID. Oh, shit. That's... Can you do one thing? I have forgot to mention about the reference ID in Google form. Can you do that? Just write down a reference ID of your particular practice.
Okay, ma'am.
Okay?